All right, in this video, I'm going to go over the basics of genetic engineering. This is what we talked about in our class meeting, but I just figured I'd make a separate video to summarize because why not? Nothing better to do. Just kidding. Trying to get my likes up on YouTube. If anyone wants to make a title thing for me with maybe a song, I'll give you extra credit. So, because trying to be famous, remember? All right. So I'm gonna just go over the slides from the meeting. Okay. So genetic engineering, basic overview, I'll go over the diagram. The definition is that it refers to the direct manipulation of DNA, which we refer to as genotype, to alter an organism's characteristics or their phenotype in a particular way. So just like regular engineering, genetic engineering, you're building something, you're modifying something with the idea of making it better or improving it in some way. So we also refer to this as genetic modification. When we talk about genetic modification, they refer to this as GMOs, genetically modified organisms. A lot of our crops are genetically modified. There's a lot of debate on whether this is a good thing or bad thing, but the science generally says that in terms of eating genetically modified food, it's not, dangerous to your health. Almost all the corn and soy products in the US are genetically modified. And the reason why scientists have started genetically modifying food is because with the idea of like these crops could be more resistant to insecticides or pesticides or herbicides, and maybe you can grow crops that are cheaper or making more of it to help people who may not have as equal access to food sources. So there's a lot of debate, but if you look at this wrapper, a lot of our food is now labeled with this right here. So if it is, if there's GMOs within it, they'll label it, so it says partially produced with genetic engineering. In this case, I believe it's a cocoa bean, so the chocolate. In this chart right here, it's just kind of showing you the same thing I just said. Most of the like soybean, cotton, and corn that is grown in the US is genetically modified. And the HT and the BT on the bottom, it refers to herbicide tolerant and also insect resistant. So just something, if you're eating corn, probably genetically modified. Okay, so going back to the diagram. So this diagram, I took it from yourgenome.org. The website has a lot of good images and summaries. So if you want to like read a little bit more, go there. So I'm going to start with number 1, which is our bacterium or bacteria cell. They are prokaryotic, and bacteria cells, just as a recap of prokaryotic cell structure, they contain one circular chromosome. So this is the chromosome, and then they also have plasmids. So these plasmids are extra pieces of DNA. They replicate by binary fission, which is mitosis, so they just make clones of themselves. In theory, they're pretty much the same when they divide. So a plasmid is a small circle of genetic material that replicates independently. So these plasmids can replicate and make copies of themselves within the bacteria. They are used as vectors to transfer genes between cells. So the term vector is kind of like sending or movement. So these plasmids, they can pass on plasmids between bacteria cells, or bacteria cells can take them in from the environment. And this is good because it allows for more genetic diversity within bacterial cells. Because they reproduce through binary fission, they're all going to be the same. And you don't want things to always be the same, because if something, disease comes or something comes, it wipes you all out. So that's not good. But with these plasmids, it gives bacteria a little bit more diversity. So it's good for them, but bad for us, because this is one reason why we have antibiotic resistant bacteria. So with the idea of genetic variation or diversity, maintaining variation in prokaryotes, they do this in three ways. So the first way is transformation. Transformation is naked DNA uptake by bacteria from the environment. So the idea with transformation is if there's bacteria in the environment around a bacterial cell, if there's DNA in the environment around a bacterial cell, these bacterial cells could take it in, and then the DNA that they take in can give it the bacteria new traits. We also talked about this when we were learning about the historical experiments with Griffith. He kind of found out that bacterial cells can be transformed. The pneumonia cells went from like non-dangerous to a dangerous strain because of the DNA they picked up from their surroundings. Another type is transduction, which is bacterial DNA is transferred by viruses or bacteriophages. So if a phage picks up a piece of DNA from a bacterial cell, and then it releases it into another bacteria, this new bacteria can take up that new DNA, insert it into its genome, and then gives it new phenotypes. And the last way is conjugation. So this is DNA transfer between bacterial cells. In this case, a plasmid from one bacteria can be transferred to another one. So they physically will connect to each other. They also call it like bacteria sex. It's like they connect to each other and they transfer DNA between the cells. The last way that there's variation is because since bacteria reproduce rapidly, so they are always reproducing, replicating, every time you replicate, there's always a chance of mutation. So bacterial cells can get a lot of mutations, and that will give them variation. All right, so with genetic engineering, we're gonna mostly focus on this top one, transformation, on the top. All right, moving on. Step 2 is the idea of cutting open these plasmids with something called a restriction enzyme. So restriction enzymes are enzymes that cut a plasmid at a specific sequence. We call this a restriction digest. So they cut DNA at a very specific sequence called a recognition site or a restriction site, or we also call it a cleavage site. Cleavage, remember, means cut. So these restriction enzymes will cut at a specific site. On the bottom image, I gave you an example of a restriction site. So if you look at the top row, it says GAATTC. On the bottom row, but going in the opposite direction, it also says GAATTC. So this particular restriction enzyme called EcoR1, it cuts at this sequence. So every time it sees this exact same sequence, it's going to cut it along this line. And it only recognizes this sequence. When it gets cut, you end up having these two pieces of DNA with some bases that are not paired with anything else. We call them sticky ends. So this is an example of a sticky end. So you have AATT. They're not really going to be base paired with anything else. And then this bottom one, TTAA, also won't be base paired with anything else. But if they come in contact with each other, they'll want to match up. So that's why they call them sticky ends. You can also create blunt ends. So on this diagram, some restriction enzymes will just cut straight down. So wherever these arrows are, that's where it will cut. So this first enzyme will just cut between the G's and C's. So we call it a blunt end because it's just going to be straight. The other ones on the bottom, this dotted line, they're showing you that this would cut at in a jagged way, so you would produce sticky ends. So restriction enzymes will cut the plasmid open on this diagram. So number 2, cut the plasmid. The purpose of this is so that once the plasmid is cut open, you can insert whatever gene that you want to produce. So in this example, we're trying to make insulin. So insulin, people who are diabetic, they need to have insulin. And so one way that scientists have figured out how we can get more insulin is to insert this insulin gene into a plasmid and basically ask bacterial cells to produce insulin protein for humans to then package up and use. So insertion, in this stage, the gene that you want to add is inserted into the gap in the plasmid. The gene that we want to add, we usually call it the target gene. So on this diagram on the bottom, we have the target gene. And then you're able to stick this target gene within the plasmid. So if you cut using the same restriction enzyme, you'll have the same sticky ends. The bases will be complementary and match up. Ligase is an enzyme that is used to seal the DNA together. So if the target gene is cut by a restriction enzyme, you have these sticky ends. If the plasmid is cut by that same restriction enzyme, you have the same sticky ends. So this AATT on the target gene is going to want to come in and base pair with the TTAA on the plasmid. And on the other side of the target gene, same thing. These bases that aren't really connected to anything right now are going to want to base pair with these bases. So you'll end up having a plasmid with now your target gene inserted. So once you have this plasmid with your target gene, we call this a recombinant plasmid. So the idea is a recombinant plasmid has DNA from two different sources. In this case, the yellow is from the bacteria, the red is from humans. Step 4 is we want to get these plasmids into the bacteria. So this is called transformation. The plasmids enter the bacteria cells through the process of transformation. So as a recap of the earlier slide, transformation is when the bacteria cells take in DNA from the environment. So these plasmids are just hanging out in the environment around the bacteria. Sometimes these bacteria cells will take that DNA in. The last part is once these bacteria cells have the plasmids within it, steps 5 through 8 is the protein production. Really, we're asking these bacteria cells to become little protein factories, little insulin factories for us. So this is now our genetically modified bacteria. We have engineered it to now produce the protein, which is these blue dots. And in steps 5 through 8, the transformed bacteria replicate and they produce the desired protein. This step, generally, we can refer to it as cloning. So the term cloning is making copies of something. So bacteria cells are making copies of themselves, right? So because they're replicating. But through this process, they're also making copies of the plasmid. So the plasmids are also kind of replicating throughout all this. So you have multiple plasmids, you have many bacteria cells, and now you have a little insulin producing factory. So this is possible because the process of protein synthesis is pretty much the same in all organisms. This means that bacteria cells, human cells, we have the same genetic code. We have the same, like DNA is still GCAT, RNA is still GCal. And so bacteria cells can produce human proteins for us. This is, so the genetic code, when I say it's universal, like pretty much all living things use the same genetic code. We call this a conserved process. So it's evidence of evolution, evidence that everyone evolved from a common ancestor because we have these shared processes, things that are the same within all living things. Moving on. So when the bacteria, they're producing insulin protein, steps 6, 7, and 8 is just showing you that once, as the bacteria are producing this protein, the protein will get purified and then packaged up, ready to be sent off to patients. So this is a general overview of genetic engineering. Here is, just as practice, okay, so here is the matching A, each of these letters. So A through, it's a little bit out of order, K is up here, and then here are your options for the vocab. All right, so those of you guys who did this with us in the meeting, I posted the answer key, but I'll also show you the answer key in a couple of seconds. But pause if you need to try to do this on your own first. And then make sure you tried to do it. And here are the answers to the matching. Yes, and so here is how this would match up to the diagram. Another thing that you can also practice, just as a review, is to summarize the big picture genetic engineering in complete sentences using this diagram. All right, so here's a simplified diagram of genetic engineering. And I put on the bottom some vocab that you could try to use to explain what's going on. All right, So this video was an overview. The next video is more detailed about plasmids and transformation. And then the third video is going to be a big picture overview, walking you through our Amgen lab that hopefully we will do when we go back to school. Right? Let me, this.